everybody. My name is Natalie Yeadon. I'm the CEO and co-founder with Impetus Digital. At Impetus Digital, we have built some of the best-in-class asynchronous and synchronous virtual collaboration and communication tools. We have worked with life science companies from across the globe over the past 13 years to help them with everything from virtual advisory boards, online medical education, online investigator meetings, and everything in between. And since the launch of our award-winning Insight Events platform, we've been helping pharma companies as well with their MSL training, corporate events, innovation hackathons, and all kinds of other innovative programs. But more importantly, at Impetus, we really believe that everything starts with a conversation. And from these big, hairy, audacious conversations with some of the leading edge thinkers, the digital provocateurs, the healthcare leaders, is we can all work together to collectively and positively disrupt healthcare. So I'm super excited to having one of these thought leaders at the table with me today. And this is actually Jessica Schwaber. She's actually the scientific director and Toronto site head at Evectus. Starting in the utopian village of Arden, Delaware, she has always been caught up in the thrill of creating something new. At present, that includes progressing gene-modified cell therapies. As a chemical engineer with a PhD in biomedical engineering, she aims to integrate a broad skill set to find systems perspectives that yield very unique opportunities. Welcome, Jessica. So happy to have you on the show today.
Thank you, Natalie. It's an absolute pleasure to be here, and thank you for having me.
Absolutely. I'm always thrilled and always honored when I have other women at the table with me going into the STEM area. We oftentimes in the traditional area, we haven't really seen that as many. I'd love to get an understanding from you what career trajectory you chose and how you landed in the position that you are in today.
Of course. So as we're talking from a professional journey, I'll start with graduating from Smith College. It's an all-women's college. And I graduated with a degree in chemical engineering. So thereafter, I joined a startup company. Back then, it was a push to join the Archon Genomics X Prize. I don't know if you're familiar with it. 100 genomes in 10 days or less for no more than $10,000. It was a big deal then. It may seem obsolete now, but it's a big push at that time. So this is my glimpse at that time of really getting into that world of cell and gene therapy. And I realized I wanted to be a part of this world, but with the degree in chemical engineering, I lacked a lot of the skill sets or toolkits to really effectively join that. So I looked around and I found a professor. I had a lab, and he specialized in exactly the areas that I had identified as gaps in my knowledge. The only problem is that he was based in Australia. So of course, in today's world, it's not a huge barrier. So I packed my things and I went and joined Lars Nielsen's lab. He's at the University of Queensland. And I gained that skill set that I'd set out to learn and really enjoyed my time both in Australia and in his lab. It was a whirlwind of a time. But of the things that I loved about Australia, it was a slower pace of life, really being off the beaten track. That frustrated my husband. He really wanted to be in the heart of the action in North America. And so needless to say, that the next stages of my life were pretty simple. I would say followed my husband simply back to Toronto, Canada, where he now laughingly teases me because it was a fantastic decision. At the time, I may have been cranky about it, but it's worked out wonderfully. And we both moved into working as development engineers. And we joined a company called Center of Commercialization for Regenerative Medicine. It's CCRM, it's a CDMO, which is located in downtown Toronto. And this took me on the journey from working at the small single cell level, in which I was working with sequencing and modeling based techniques, into more of the therapeutic world, really working with process development. And that involved scaling up, closed systems, high throughput analytics, things along these lines. But most importantly there, I'll say, is that I met my core group of people, brilliant, driven individuals. And they're both my social circle and my professional network today. And from there, I moved on to AVROBio. It was a rare disease therapeutic company. And they were just starting to build out their manufacturing for multiple different indications. And this really appealed to me because you can learn more about the final stages of the process of a therapeutic at this point. So you can have the craziest ideas during the R&D phase, but without really understanding that final point they have to get through in manufacturing, you can not really understand how to develop a product properly. So that brought me to my role now as the scientific director in Evectus. And I would say just for that last transition, as you can tell, I love to learn a lot about new different processes, gaining the large picture on things. And I found a position was open with Evectus. And I was intrigued to understand the therapeutic development world from the angle of working as equipment or a piece that would support the therapeutic world rather than developing a therapeutic itself. And additionally, they were looking for somebody to build out the Toronto site. And one of the things that I believe I'm okay at is really connecting people and ideas to make new ideas or to meet a need. And I wanted to apply this skill set to a small hub, and it was especially appealing to the growth potential that Toronto holds.
Beautiful. I love it. So let's dig into Evectus a little bit. What is the problem that Evectus is trying to solve?
So Evectus is developing a delivery technology platform. And so this is to enable gene editing of a cellular therapeutic product outside the body. And so you're trying to deliver cargo across the cellular membrane so it can integrate that cargo. And we're trying to do this in a gentler approach compared to the current methods available today.
And so so let's actually dig into that for people just kind of listening, because some people may not be as scientific. So what is the current way that we're delivering medication?
So the most common way, which is well recognized today, is using viral vectors. And so it's hijacking basically a viral machinery or capability for delivering cargo to cells. But instead of infecting the cells with what the virus would use to replicate itself or create more of itself, it's delivering a cargo that we want it to deliver to then integrate into the cell and transition the cell's functionality and whatever means related to the cargo that we deliver. So it could express a new protein, which could target certain cells, such as a cancer cell, or it could prolong a cell's capabilities, driving a functional phenotype, pushing a cell to behave a certain way that it may not otherwise.
So this is the way that we've been doing it. We've been using viral or non-viral technologies, basically injecting something in and creating the cells to make something for you. What is some of the disadvantages of that methodology, basically the problem statement, and why you are perfecting that process or making that better?
Of course. So the viral technology is it's been used for a long time, and it's a fantastic approach. I would say from a safety viewpoint, it can integrate into anywhere within your genome. Comparatively, if we're looking at something along the lines, another common method used is electroporation. It induces electrical current to the cell to be able to temporarily disrupt the cell membrane and allow material to transfer into the cell. Now, of course, cells don't exactly appreciate being electrocuted. And so what we find is that these cells are less healthy post-processing and can even result in them not functioning as well. They often don't divide afterwards, or they may not actually carry forward the function, even though the cargo may have been delivered to them. One other drawback, just to touch on in terms of viral vectors, is that they can only deliver any sort of nucleic acid cargo. And so when you're also looking in terms of that, it limits the capabilities compared to what we're developing. You're able to deliver everything from nucleic acids to small molecules to proteins, even including what's really popular today, which is Crispr. And I'm going to touch on one more thing, Natalie. The one more thing is manufacturing. And so if we're talking about using viral vectors, because it's so popular, the manufacturing is actually backed up. It's quite expensive. It's quite hard to get your foot in the door in that space right now. And so there's also limitations if you're trying to develop a therapy very quickly now. It's hard to get your foot in that space. Sorry.
So tell us about the cell engineering technology platform that you are creating at Evectus. It's the solute. So what exactly is that?
So the solute, it's great that you've looked into Avectas in this way. Solute is actually the process. So the process is, again, temporarily permeabilizing the cell membrane. And it's using a mechanical spray to deliver the solution, which could contain the cargo. Again, this can be nucleic acid, or this can be proteins, small molecules, and that's delivered by literally spraying this onto the cells and permeabilizing the membrane for that short period of time where the material enters the cells. And in the cell membrane, can reclose over, and the cargo has been integrated into the cell. We've seen that this is a gentler process. So basically the cells retain functionality afterwards, they divide well afterwards, and what we can see is that we're looking for them to have better functionality as well.
And what about things about cell proliferation? I mean, this, comes down to the whole question around being able to, augment what you're doing, or replicate, or, to maintain or build from like problems that are happening with organs and tissues and what have you. So if what is the benefit of what you're doing in terms of that? Also things like questions around manufacturing timing, and also things like cost to people who are trying to do some of this delivery.
Of course. So one of the things I would say in general, just this ability for a cell to divide after you have perturbed it in any way, it shows a cell health. And so it shows that once you've transferred this material, let's say to the patient, that it will continue to divide, proliferate, and carry forward that edit which you have added to the cell. You don't want the cells to just be finished, for example. You don't want them to not be able to carry forward the edit, expand, and continue to be efficacious once it's been delivered to the body. Comparatively, if you have a therapy and you add it to the body and they die, either in the process of thawing or transferring them to the cells, of course, they aren't going to target what you have designed them to target. And so if we're talking about other things in terms of phenotype or aspects along these lines, you may have to develop fewer cells. And in the cells themselves, you wouldn't need a therapy right now. We're looking at delivering billions of cells because we aren't quite sure what works at this point. We're early in the stages, and so everybody's trying to figure out what cell type works, maybe some cell types that work or combinations thereof. But right now we're just throwing the kitchen sink at it because we don't know at this moment. If we can get down in our understanding to fewer cells and have them divide within the body, it can be a more efficacious and more appropriate cell type to be delivered to the body. And so what you want to do is maintain that functionality, potentially deliver less cells, and have a more effective therapeutic. In that regards, cost as well. Of course, larger cells, more expansion time, et cetera, that can really build up in cost. And as for us, in terms of our timing of our process, it's actually quite fast. We have a closed GMP system, which we're looking to have come out next year. And it can be hooked up to any sort of closed cell culture system. And you would just transfer your cells from that system into the, it's basically a single use system, which would be applying the solute process. And you would go ahead and edit these cells and then harvest them back out to go back into whatever downstream application you're looking for. And that can happen within a very quick turnaround time. And so that's, again, the timing on that compared to having to wait for an LV virus, which you would maybe have to wait for a period of time to get the virus in hand before you can even develop it. The solute system right now, and as soon as 2022, you would be able to start working with the therapeutic systems.
So what exactly is the business model at Evectus? Are you a service company, or are you selling a product to the end user who is then going to utilize your technology?
What we're looking to do is work closely with companies in developing their therapeutic. We don't necessarily want to just be selling the equipment and then consumables thereafter. We'd like to work with a company in developing their therapeutic side by side with them.
And we hear a lot about things, recently the mRNA. I mean, everybody's heard of this because of COVID-19 and the vaccine.
Of course.
So with this technology, could things have been different, or could you be building vaccines in a different way in the future?
Well, with mRNA, so you can transfer mRNA to cells to get a transient effect on them. And our process, the solute processing, can transfer mRNA as well as other nucleic acids. The idea with that is you aren't making a permanent edit to the cells. You're getting a transient effect to, therefore, not to, of course, have an immediate effect on something, but it won't be lasting. And therefore, there's seen as a level of safety with this approach.
You were also mentioning that people are utilizing the solute process for Crispr and also probably Cas9. Maybe you can describe to the listeners what that is exactly and how you are augmenting the process?
Of course. So we aren't actually augmenting CRISPR Cas9 itself. It's more just the approach in delivering it.
Right.
But that approach within itself, so it's like many things, we hijack elements from the microbiology field. We find that there's a lot of sharing of different DNA elements between microbes, and we find them to be incredibly useful, and we find that they work very well with our own systems. And so similar to what we've seen with the virus, we've modified many of these systems to be able to work within our own primary human cells. And so CRISPR itself, we have taken a protein and a small piece of guide RNA, which will be able to locate a particular location within your DNA to make an edit. And so comparatively, when you're saying viral vectors, for example, can place an edit in multiple different locations, this is really guided in terms of where it will make that edit.
How will your
targeted?
Yeah, it sounds like amazing. How will your technology help with things like T cells and NK cells? I was thinking specifically about immuno-oncology. What is some of the benefits of this technology?
Yeah, you've hit it spot on. That's exactly the cell types that we're working with. So T cells and the CD19 CAR have been the primary focus of the cell and gene therapy space for a long time. And we're starting to realize that there's multiple other cell types that could be efficacious as well. You don't have to just focus on the T cells. There can be NKs, macrophages, and we're even starting to take subsets of T cells such as tumor infiltrating lymphocytes. And so as you start to expand the cell types that you're interested in, we find that the T cells themselves are a good model to start with in terms of showing that not only can we work with T cells and make them have the same phenotype post-processing as unmanipulated cells, but plus the edit that we've introduced. We also want to show that those cells will maintain phenotype, expansion, et cetera, to the non-manipulated cells, but carry forward that edit. Now, that's just one way of improving a process that's already being done. Another thing, though, is there's many cell types which may be more fragile. And so you mentioned one there is NK cells. And as those may be more fragile cell types, some of these other approaches may have more deleterious effects on that cell itself. And so what we want to do is show that using our gentler process, you can result with more cells in the outcome. And so not only improving processes, we're adding abilities to the more fragile cell types as well.
Amazing. That's really interesting. Thoughts around allogeneic therapies. What can you do in terms of that area around, especially around post-transfection expansion steps, and what you sort of do to change that system?
Right. So allogeneic therapies, as you know, are becoming a larger and larger field. So depending upon the approach, I think the most common one people are talking about these days is starting with induced pluripotent stem cell. And so by using iPSCs, just the acronym for the induced pluripotent stem cells, and working with those cell types, one of our biggest approaches there would be able to introduce an edit early on in the process. And again, because we aren't perturbing the cells as much in terms of their primary functionality or genetic state, we can maintain a higher level of stemness. This is the idea, is to maintain a higher level of stemness in these cells after they've been processed, meaning you're not shifting or sorting through all these cells at the end of the processing and discarding all these clones, which may have been perturbed in ways you don't want. And you have a higher number of clones which can be used moving forward. And so as these cells have been perturbed less, it's less effort to be able to sift out those specific clones you want to move forward with. And then I would say in general with allogeneic fields, and if you're trying to knock out specific elements, for example, B2M or things like this, which would maybe create any of these graft versus host or host versus graft diseases, I can easily introduce, again, that CRISPR to be able to knock out genes of these sorts.
Wow, fascinating stuff. And I'm sure that there's probably some work you're also doing in multi-dosing. And I'm thinking specifically about the more popular things around CAR Ts and CAR NK therapies. So what kind of work are you starting to instigate there as well?
So the CAR NK therapies, we have multiple different, again, just trying to deliver our different CARs to the NK cells. So we were working with delivering CD19, let's say, to NK cells and seeing again that they may be gentler and maintained on the downstream post-processing. You're trying to get high recovery, high efficacy of these cells after you've delivered any of these CARs to these cell types. So for us, again, it's a method for being gentler, not perturbing these cells as much, and maintaining high viability and functionality post-processing.
So pharma companies are working with you, wanting to sort of establish some of this, more gentler way of maybe doing some drug discovery. How do you think, sort of in the broader picture, how do you feel that a technology like yours is going to change or potentially transform the way new drugs are discovered?
So I would say in general, it's a hard question because at this point it's a hard question, the fact that it can go so many different ways. And so I would say in general, when we are looking to enter this space in terms of making a true impact on this space, we again are trying to identify exactly these cell types which may not have been used before, or we're not trying to identify, but being able to work with these cell types which may not have been able to have been used before because they weren't able to work with the harsher systems. So if we're talking again with electroporation as a general approach for delivering cargo that's non-viral, what I would say is that many cell types are not able to withstand the electrical field or pulse, which is implemented on them. So for us, it may allow that door to be opened just to other previously non-explored cell types.
And we hear all the time about the word precision medicine and, uber specialized N=1 medicine. Are we now heading in that direction with technologies like yours at Evectus for coming up with these super specialized medications?
I would say that we've been there for a while in terms of considering this precision medicine. And what I would say in general is if we're looking right now with using autologous therapies, that's dominated the space. And I would say that is very individual based. You're taking material from a single individual, you're modifying that material, and then you're returning it back to the same individual. This is not cost effective. It's exhausting in terms of the process variability and trying to work with one person to another. I would say that one of the things we can work with, again, you touched on this with allogeneic work. And so what we'd be looking at there is trying to start with a material which has been highly characterized. And then if we're talking about precision medicine in terms of particular targets to be hitting for somebody's specific cancer, that can still be introduced at the iPSC stage, that can still be introduced to, then the process is just a slightly different edit on that point. In general, I would say the field right now, if we're to talk about solid tumors, for example, and we're looking at something called black antigens or dark antigens, what we're looking for is trying to identify new antigens which may not be previously elucidated or understood that are expressed on the cell surface of many of these cells within these solid tumors. And they're trying to find targets that are broadly expressed across cancer types to then target those across a broad amount of patients rather than having to specify particular targets for particular patients. But precision medicine will always be there. We just need to figure out if there's broad targets that can be hit. Or if we're going to
So what's really interesting about the conversation, Jessica, is that technologies like yours are so incredible and are going to do so many amazing things. On the same side, you can probably hear the other side of the story talking about the expense of the healthcare system and how, these costs are going to become exponential with these very complex medications, therapeutics, and a whole assortment of forms and functions. How do you actually answer that question as we get into this almost must-have level of medical complexity and be able on the same side to say, how do we actually manage the cost of healthcare?
Yeah, so these, as you mentioned, these costs are not sustainable. And so I think this does come down to what we were just talking about with the autologous therapies. You have small batches of production, and these have large costs that are due to QC and labor requirements. And so QC quality control. And so what you're trying to do is look at a lot of these assays and make sure that your product is performing the way you expect, but off of a lot of multiple, very small doses. I would say in order to improve these things, you're really looking to increase the therapeutic batch sizes and you're trying to automate wherever possible. And so that's talking about scale up and automation.
Love it.
That-
And it's so absolutely essential. Please go on. Sorry, I interrupted you.
No, that's it. Go ahead.
Yeah, I think it's really interesting because I like the fact that Evectus is not just selling technology, and like you said, other sorts of components that sustain that, but you're also looking at true partnerships. And, we know that there's a legacy process for doing new drug discovery, and that includes, INDs and BLA filings and regulatory this, that, and the other thing. My curiosity lies in is now that we're in this new post-COVID world and we've sort of seen regulatory kind of go on top, get turned upside down. Do you sort of see the need, especially with technologies like yourselves, where there has to be almost like an innovation or a rethinking or a disruption of the traditional way we've been doing clinical trials or the whole regulatory space?
Of course, I think in general, even the current release criteria that the FDA are using for the cell and gene therapy space will quickly become outdated. And so right now, I think the FDA and the cell therapy world, they're both staring at each other, trying to determine who's going to dictate the acceptance criteria for cell therapies. And the truth is, we're just trying to figure out the edges of the space. It's very hard to figure out what should be filled in between. So for example, if we're trying to talk about characterizing our cell types, if we talk about a classic assay, potency, how do we define potency of a cell therapeutic outside the body? And one of that has been a very hard model to define. If we're also talking about long-term side effects, that's only going to come with time. We can't speed that up. And compared to small molecules in general, you're trying to characterize a cell, which is incredibly difficult. We can't break it down the same way and say exactly that it consists of this, let alone the heterogeneity we see within single cell types today. So I'm not sure exactly if this would break it down any positive, faster way with cell therapies. What I'm saying is that it's a complex road and nobody knows exactly at this point where it's going to take us.
Will these technologies change the way clinical trials are done? Currently, many of them are done in patients. We're starting to see people backtracking and doing more trials in silica and what have you. So is there going to be a world in which all of this is going to be done outside of patients?
I mean, that's very forward thinking. I love it. But I would say for myself that I guess that comes down to your precision medicine aspect as well. And it comes down to the potency assay, the need to really develop a potency assay and understand safety. So if we're talking about phase 1 clinical trials, you're looking for safety. And then from there, you're trying to build up your efficacy and seeing how well your drug therapeutic is going to be performing. At this moment, I would say it would be fantastic to be able to truly determine this outside the body. I do think that that would be capable, but it would take a leap of thought and it would take a lot of dedication to that.
What else do you think are current barriers to fully realizing the potential of cell and gene therapy? What's your thoughts there? What needs to change?
I think it's two-pronged. I really believe that it's both conceptual and technical. And so if we're talking about working conceptually, you're both looking at trying to find universal and effective targets. So if we're thinking about CD19 as a CAR or a classic example of CAR, we learn that targeting this molecule is not completely effective in the fact that it doesn't always fully eradicate the cancer. There's still going to be several cell types that remain and therefore not fully eradicating the acute lymphoblastomic leukemia. So focusing specifically on the CAR, we know that we may need to identify more molecules like we were talking about with the dark antigens, or we need to identify stronger CARs. And of course, this is going to be learnings that build upon each other. And mostly I'd also point out, like I said, that biology is complicated. So your single cells that we talked about are heterogeneous. You're going to have multiple different cell types you're trying to scan across. And often you aren't looking at a single gene, but rather a polygenic system, the networks that are working together and developing in space. So first is characterizing the disease we're trying to target. And then if I shift to technical, there's a whole host of issues as we briefly touched on before we were saying, can we do this outside the body? I love the idea. I honestly had not made that leap of thought, and I love the ingenuity behind it. But currently working within our systems here on, not to say here on planet Earth in 2021, unfortunately, we're very early in the stage of producing these therapies. It's challenging, it's hard to scale, it's hard to characterize, and it's hard to truly create these allogeneic therapies, as we touched on as well. So scalability is a problem. Defining our drug product in terms of characterizing it is difficult. The FDA has some very basic criteria really focused on safety, which is critical, but again, lacking a little bit in defining that potency. And so I do believe those are elements as well, which are huge barriers. And then lastly, there's cost, which we touched on. And I believe that will come through manufacturing and automation as well as scaling these systems. So we have equipment and companies such as ourselves, where we're trying to create solutions, trying to build manufacturing steps. So for example, you can have one system which would hold all processes within one piece of equipment. You'd have other companies that approach it by building several unit ops which tie together. And with any of these systems, some will boil down to simplicity and others come down to agility, but they all have their benefits and drawbacks. I just think we're in the very, very early stages of this.
Which is very exciting, being a pioneer in that area, Jessica. And, you know, I love the fact that you're located in Toronto. So Toronto is known for many great things, including our artificial intelligence sort of ecosystem. I'm actually curious if AI is being built into your system somehow. Are you leveraging artificial intelligence?
We are not introducing that, but do you have any ideas on how we should be or could be?
I just love the idea of machine learning as you're starting to learn about, you know, the various methodologies and just making your system smarter.
That makes a lot of sense. I mean, we are capturing data of how the system works and using it to improve our system, but we aren't necessarily including AI at this point.
Yeah, you can feed it into an AI machine learning system or in an algorithm, and it might be able to potentiate, you know, speed or, you know, other things as well. So in the drug discovery process. So really interesting. I love also the fact that, you know, we're talking about partnerships. And so that's a very much a big part of the je ne sais quoi behind your company. I really love the fact that you're doing that. Can you tell us a little bit about what you're sort of doing with the CCRM?
Sure. So CCRM, first of all, we're located in the Mars Discovery District. And so it's a large glass tower, which is right downtown in Toronto. You have University of Toronto on one side, just to the north, and then to the south and kind of surrounded you by core hospitals. So it's really right in the center of it all. And within this building, we have CCRM 3 floors down. And so CCRM is a CDMO or a commercial development. They help us continue to develop a process and investigate different work packages that we put together to gain learnings on how our process may be operating. And so we had several bodies work with them. And one of the reasons for moving to Toronto is to be close by to them. So they've been a great collaborator in that regard.
Yeah.
And I would say that in general, they have an incredible suite of equipment. I have never seen quite that level of equipment in a downtown space. It's fantastic. And just, I mean, there's so much in Toronto. It's really becoming quite the biotech hub. It's shocking how much it's grown just since I've been here in these last 5 years. And we have Sunnybrook Research Institute just north of us, surrounded by, even within our space itself, it's just filled with startup companies. So the connection among companies, and you even have a group, Medicine by Design, which organizes talks to really bring in you're bringing in multiple different academics to the world too, and trying to make that bridge to industry. It's really quite an engaged and innovative field in which people are trying to truly make those connections happen.
I think a lot of people are talking about replicating the biotech haven that's been built in Boston. I know that, you know, the leader of Moderna, you know, graduated a long time ago at University of Toronto and was kind of bringing back some of that thinking. So I think it'll be very interesting to see what happens in the next 10 to 20 years here. What about the partnerships with Vicelix? What are you doing with them?
Of course. So first, one thing I will say about Toronto that is a leg up compared to Boston is we're a highly international city. And so there's a
That's
huge
true.
draw for international talent in biotech. And I think it's been stimulating just to see what people are bringing. It's a very, very international city comparatively. It's
Absolutely. It's really, really
um,
refreshing.
so with Phycelics, I would say they've been another one company that we're working closely with to deliver cargoes using the solute platform, and has been a great collaborator in terms of trying to develop the system and be able to, again, looking to slot in the solute technology into their manufacturing process.
And ONK Therapeutics.
Yep, you're going, like, you have done an incredible job of really, really looking into our background. So there's several different partner groups that we are working closely with. And what we're looking to always do is we're looking to continually build out with different partner groups what different cargo they may be looking at, what different cell types they may be looking at, and how to develop our device to best meet those needs. And so some of these that you've mentioned, we've worked with Phycelics, we've worked with ONK, we're working with many other partners to be able to see how well our system can fit into their manufacturing process and meet their needs, but of course, adding that effect of having a gentler process. And
And
so,
I love the work that you're also doing with J&J Innovation Lab. So maybe you can speak to that as well.
sure, so we're based out of J Labs in Toronto. And so what we're working with there is, it's basically an incubator hub space. And what we're looking to be working with there again is, it's a small space which you rent out, and you continue to have a place where you can really engage with other startup companies. So that's basically what's so great about being in the downtown Toronto area, is you have a true lab that you're able to not only immediately engage in, you don't have to worry about the full setup or the infrastructure, it's there, you just move into this incubator space. But I would say some of the other things that have been incredibly fruitful have been some of our work with academic collaborators. And so we've been working with the Karolinska Institute and aspects along these lines in terms of being really engaged in developing our capabilities in the NK field and how to continue to work with these more fragile or gentle phenotypes.
And I'm assuming a great collaboration with the UC Davis, I guess, University of
Incredible group
California
there.
as well.
Yes, it's been, and that's part of the reason for being in the North American hub. So being located in North America is you're very close and you're very central to many of the different partners and many of the different companies that are based in this immediate region.
So is your company now have a viable product? Are you profitable? Are people still investing? Is there going to be an IPO? Where's things at from that standpoint?
Yeah, so in 2022, we're going to have this GMP system that's available. And so at that moment, we are looking to start, we're still engaging with partners in the meantime. We have a research tool. It's exactly as it sounds, it's for research. And the idea of this tool is to be able to have that be a stepping stone to working with the GMP device. And so we have that placed at 10 different partner sites or collaborators. And the idea is to continue to use that to look into these different partnerships and to be able to continue to fuel those relationships as we build towards that GMP system.
And what kind of approvals are you getting? And do you have to do trials, or what is that looking like? I know scientific papers and journal publications on your results. So tell us a little bit about what that plan is.
Yeah, of course. So we have a publication that just came out earlier this year, and that's showing some of our latest capabilities and some of our latest experiments, more from, again, delivery of different cargo types to different cell types. And what I would say in terms of our work that's ongoing, we are working with multiple different vendors and multiple different groups to be able to really narrow in our production line to meet those GMP specifications. And we also have, of course, you generally work with a very small company. We're 40 people based out of Ireland. So it was built out of the National University of Ireland, and there's 25 people there. There's a small group within Toronto, there's 4 of us here, and then a site in Cambridge. And so we're all trying to work together to make this happen. But because you're a smaller group, you're often working with multiple different partners and different groups to support on these fronts as well.
That's exciting. Your predictions for the future of cell and gene therapy and precision medicine all rolled into one ball. What is that going to look like?
So for me, I would say my prediction in general is you have two sides of the same coin. You can go both ways. You can become a very powerful tool for developing bespoke and personalized therapies. And this will be, of course, challenging technically, but I believe it's quite doable. And the other approach is going to dictate that therapies will be so general that they could be applied to any human being. And so that means that your allogeneic therapies that we've been referring to will fly under the immune system radar and have the same effect across all human beings. Regardless, I'm hopeful for the future. Obviously, I think that costs will be reduced, potential risks understood, and therapies will be applied to earlier and earlier stages of disease. But those would be, no matter what, it's the flip side of the same coin. It just matters which approach you're going to go for.
I love it. I love it. Pharma companies listening, especially in the scientific affairs, drug discovery area, what would they do, or how would they approach you for partnerships? What would that look like?
I would highly recommend that they would be reaching out to our business development team. That's based out of our Cambridge group, but anyone, if they just reach out or go to the Evectus website and were to get in touch, we are consistently reaching out to different partner groups and looking to build such evaluations and such research agreements to work with people.
Very exciting. So this is a great way to wrap up. It's a great, great company. I'm super excited for what you're doing. I think it's really leading edge. For those people who are listening, who are intrigued, want to learn more, see if there's some sort of a partnership or collaboration experience. They're located in the Mars Discovery District in Toronto, downtown Toronto. They'd love to hear from you. We will be leaving Jessica's contact details in the show notes below, so please be on the lookout for that. If you enjoy this conversation, we encourage you to check out impetusdigital.com. These are the kinds of conversations that we have in our Insight platform, where we work with payers, physicians, allied healthcare providers, payers, and patients, where we actually have discussions about new drug discovery, how to move the agendas forward, how to get beyond the pill thinking. And we work together through our platform through asynchronous and synchronous touch points. And we do this through our white glove service, and we help you all the way along so that you can actually get work done and move agendas forward. So we appreciate everybody if you can like and subscribe to our channel, and we would love if you can leave some feedback on iTunes. We want to thank everybody for their time today. We appreciate all of you taking the time, and thank you, Jessica, for a fantastic conversation.
Thank you so much, Natalie. I hope you have a great day.